Hello everyone, in this video I'm going to showcase how to perform survival analysis in R that using some biological genomic data. So first of all, a brief introduction about myself. I'm a biochemist and currently working on the cancer research. I'm interested to know whether a gene or its protein has any effect on the progression of cancer. Okay, this seems a little bit too general to make any research deduction. So maybe I want to ask, does the gene expression of a particular gene affect the survival rate of cancer patients? This means that if someone was diagnosed to have the cancer, will a certain gene level in his or her body tell us anything about his chance of survival? How more likely is a certain group of patients to live longer than another? I have a chart that to illustrate my question. My hypothesis is my selected genes does affect the survival probability. When it is very high, the patient can survive longer, or vice versa. And of course, we want to make a conclusion backed by a statistical calculation to know whether it's statistically significant or not. So let's understand our database first. In this tutorial, I use TCGA data. TCGA stands for the Cancer Genome Atlas. It is a collection of cancer data, a program led by the NCI and NHGRI, which are part of the National Institute of Health in US. So this is a very big project, and the project aims to identify the complete set of DNA changes in 33 different types of cancer. It's a very large project. The project aims to identify the complete set of DNA changes in 33 different types of cancer. It collects the data of cancer patients and normal individuals, that including clinical, genomic, epigenomic, transcriptomic, proteomic data, and others. TCGA data is well published. It has also some specific R package, and one I'm going to use here is called TCGA BioLinks. So which contains some main functions to analyze and convert the data into useful information. So now back to our question, which is how more likely is a certain group of patients with our targeted X gene level to live longer than another. To do this survival analysis, we just need to clarify three components: time, status, and event. Time, there is the time till an event happens, which is the overall survival time. Status, this indicates which patient have to be kept for the analysis, so whether the patient is dead or alive. And lastly, the event, which is the variable that distinguishes the status of the patient. For example, this tells which patient have the genes up and down or down regulated or have no changes in expression. Okay, let's go straight to the R analysis, and we will learn about the important terms or concepts along the process. Okay, so I'm inside the RStudio environment now, and the first thing to do is to make sure that we set our working directory in the correct location, and I want it to be in this file location. Okay, so and this tutorial I actually following this website, and they did a very well explained and detailed tutorial in getting us to analyze the TCGA data in whatever way. So we can have a look in it. Okay, so to do the survival analysis, we need the following library, and just load them one by one. There are actually a lot of libraries that can be downloaded directly in RStudio here. So we click package and install to just write the name of the package that you want to download, let's say carrot. Or some of them actually is provided by third party that is called bioconductor. So let's say survivor, you have to go to the bioconductor biomanager to download it. You can Google more to know how to install them. So in this tutorial, we want to focus on TCGA data. And so here we try to see what are the things that inside it first. Okay, so they actually contain a total of 33 cancer projects, and each provided with the project ID. So we actually interested here. I want to check the prostate cancer database. So with the project ID TCGA prep. Okay, so this data show that they are actually having total of 500 patients, and with different data categories that some of them are transcriptome profiling, simple nucleotide variations, biospecimen, clinical. So we have to see that what and some of the file actually they have more than the others. So we can say that we can see that some of the data file they might be doing more than one to the patient as compared to the other. Let's say maybe this one transcriptome they maybe did 5 times per patient, or then for clinical data there's a short kind of like 1-to-1 ratio. Okay, so now we want to focus on particular type data that of our interest. So we interested in the transcriptome profiling data, and which is like RStudio, and with the workflow type that is high state count. Okay, so we want to know it, and it takes a while for them to get the data that we want. Okay, so to visualize the query result in a more readable way, we can use the command get result. Okay, so we can see some of the column names for all the column names for the database now. And if let's say you are interested in specific things, like let's say sample type, you can just go and have a look. What are the sample type for the first stage patient? Okay, and now we want to know the class of the sample type. Oh, so this is the character. Then we want to know that we want to know the summary of this. So let's convert it to the factor category, and now we can have a summary look of the sample type. Okay, so there's only one metastatic sample. So for simplicity, we actually want to ignore the file that contain metastatic patient. So we redo the query. Try to just to reserve the other type, transcriptome profiling, RNA, but we add on the things that we want the sample type that specifically that only contain primary tumor and solid tissue normal. Okay, the next thing we can do is to download the file, so that every time, the next time when we want to use it, we can go into the file that already saved in our computer. Okay, so it will take a while. Okay, it takes around 2 minutes to get the things done. Okay, and how about we have a look at the size of the data? Okay, it's not done yet. And obviously this is a very big data, that's why it takes a long time to download all the file that we want. And this is also why we want to just get a query of the particular set of database that we want. We don't want like a big junk file of the data that saved in our computer. Okay, that it still take a while. Okay, So it has finally done, and we can take a look on the file size that actually it contains that 56,000 more of the genes that from the 550 samples. We can have a look also in the file structure of the TCGA data, and if let's say your computer is okay, you can just have a look here. So we can see that they actually contain the group of the file that in different categories, and this is a very typical example of the summarized experiment format the file. So it contains one or more essay that is metric form. So each of them, when you open it, you still can go another level of the information. Okay, here they contain three like core row range core data essay. We may use the function core names to extract the column from a given table respectively. Let's say we want to collect we want to know that what is inside the core data. So we have a look here. Oh, is that contain like more than 148 column that like inside there's a lot of information here, and we want to know like in particular maybe we want to know the vital status. Okay, what are there? Okay, and how about the tumor state? Okay, there's nothing. Okay, and how about the definition? They having two different sample type, and tissue or organs of origins, primary, glacial grade, secondary, and how about this one? The table that in the gender. Yeah, this is only male. Saying this is about prostate cancer. Okay, so how about also have a look into the essay type? So we can have a look here. Yeah, so this is all the information. We can fully explore the database so that we can get familiar with how the genomic data stored. Then from there we can have a better picture of how to extract this data for the analysis that we want to do. Okay, then we can save this data as a file, and every time we need it later or next time, once we start the RStudio, we can directly load this using this command. Yeah, and can we just start from here. Okay, so let's come to the second part of the analysis, which is about RNA-seq normalization. So before doing any analysis on the file in this TCGA data, we first have to get that data normalized first. So the very obvious thing we want to do is to obtain differential expressed gene based on some clinical phenotype. So this can be done using the lemma package. So lemma actually do a job in a way that involving linear modeling by assuming the log count values are distributed normally and the mean variance relationship is accommodated using precision which calculated by the Wolf function. Okay, and then normalize. Okay, this look very complicated, but anyway, they're actually having a lemma pipeline that help us to do the job. And the output actually is just to obtain the differentially expressed gene, the genes, and the gene signature. So here they are actually having the whole pipeline things. Okay, and so we run the pipeline first. Okay, so there are three things that to take care about. So firstly, that is about the object and the data we want to use, condition variable, the variable that we want to group the patient, and in comparison to the reference group. So we can see that the data we want to use here is TCGA data. Condition variable, we want to know to want to divide them based on the definition, which is sample type. So the reference group, we set it as solid tissue normal. let's say, so it's very handy. Just copy the function and you will do the job. If you want to know more, there's a workflow in detail for a step-by-step explanation from the website that I get the tutorial from, which is here. You can also learn from there. Okay, so in this step, it actually is a way to get a differential express gene based on the condition that we can set ourselves. So we can also set another way round, which is like, let's say we want to separate the gene expression, the genes based on the race. So we can set in a way that condition variable is race and the reference group is female. No, sorry, it should be the, like, let's say white. Oh, it's very slow. Yeah, let's say it's white people. Okay, so we can do this way. Anyway, I'm not going to run this here. And at this stage, let's tap the data to file, and next time we can open it directly from using this command. Okay, here we want to know how to get a plot of the two first principal component. It will take a while, and the plot actually want to show how we actually divide our group based on the lemma function. Okay, we can have a look at the plot, and the red color dot is referred to solid tissue normal, and black color dot are the primary solid tumor. And we can see that they actually quite well separate based on the RNA expression profile. Okay, finally we come to the survival analysis part. So let's start from the simple question. Does race influence the survival in prostate cancer patient? Remember in my previous slide that survival analysis involves three elements: time, status, and event. So let's start with extract the relevant information. So we are only interested in the primary solid tumor. Here, you only want primary solid tumor, and we want other information such as the patient ID, the vital status, so that whether it's death or life. So days to death means the number of days passed from the initial diagnostic to the death, days to the last follow-up, or the number of days passed from the initial diagnostic to the last visit. And also include here race, just optional. Okay, now we have a new data frame which called clean depth that contain the information only relevant to survival analysis. And before we proceed, we need to change the part of this information in a way that is acceptable to the method from survival package. So we need to create two more columns. The first one is that we need to create new Boolean variable that has true for that patient and false for the alive, the patient that is still alive. And also we need to create an overall survival variable that is equal to that days to death. If let's say the patient is confirmed with the status of death, or refer to the days to last follow-up if let's say the patient is still alive. Okay, let's have a look at the first September. Okay, so this is the information that inside the data frame. Vital status, and we having the last two columns, which is that we just added just now. So false, and this is the days of the survivor, or maybe the days until it's dead. Okay, how about have a look on the table? On the race. So we have like 1, 2, 3, 4, 5 races. Okay, and let's see if any difference in race have any difference in prognosis. So the survival formula is just doing in the way that we have a linear model, and in the form of independent variable to dependent variable. So the dependent variable are overall survival and disease. It means that, okay, so this one. It means that those patients with last follow-up that are part of the survival curve, but their data are only being canceled when the time of the last follow-up arrived. So the independent variable is raised. And this is the formula that we want to do the survival model. Okay, and next we can have a look of how the data show here. So they have already calculated the median, the confidence level at the 95%, and also here we can plot a Kaplan-Meier plot here. Okay, so this Kaplan-Meier plot actually shows as a different strand represented by the range. So here we have a look at the first one. Okay, so the p-value, however, it shows that the range is not significant, like in a way to affecting the risk of prognostic in this database. It's not really significant in the prostate cancer progression. And how about, okay, we want to know why that like here at the later part they actually show a very big difference, right? But how come the significant value, the p-value is just not really of what we thought? So we can maybe have a look on the patient number that reflecting that particular part. Especially we can see that is at the later stage of the time of survival. So we can see that after the 1,000 days, the patient numbers are gradually decreased, and after 3,000, we can just see the number for the black or African or white race, white species. So this is not really significant enough to show the whole picture of the survival rate. And yeah, because they are just representing a very small population at this area, and most of them are actually very similar as seen from the line here. Okay, so this is the way that to answer the simple question in doing survival analysis. Now let's look into the more complicated, more complex question, and that is about the gene expression and the survival. So now we want to ask a question: that gene expression of particular genes, let's say XYZ, affect survival in prostate cancer patients? So to do this, we actually want to do in a way use a machine learning method to classify an unseen sample as being tumor or not. We have the normalized expression value from Lima, West, Maié, and the type of sample can be found from definition, either primary solid tumor or solid normal tumor. So we want to divide the sample. We want to divide the sample, like let's say 75% that is for training and 25% for testing, to create a data partition that used for the training model. Okay, and here we want to define X train is the ID that have already divided into this 75%. Okay, and X test is the leftover sample that waiting for validation and for testing. The same goes for the Y here. Okay, so X actually means that the metric that with the normalized expression value, this is the gene expression value, and Y is the variable, is the response variable that tells us whether it's the primary solid tumor or solid tissue normal. Okay, now let's go and train the model, and here we actually use the GLM net. This is a elastic net model contain many types of the regression model, and here I want to use the one that is called binomial. Okay, so binomial is actually a logistic regression model that we have only two outcomes. So either it's a primary tumor or it is the normal tissue. Okay, so this model select the gene or group of the gene that best predict each of the condition, and use this to build a model. And then this will be used for classification. We can then look at these genes individually to see if any interesting genes or biological relevant for the classification problem is selected. Okay, so the training has finished. Now we can test whether it is actually good or not. We can using the confusion method. This is a simple table that compare the prediction. So we can see it. Okay, so the simple table that should show the two positive, two negative, false positive, false negative. And let's say you see the test sample show that the 122 is primary solid tumor and same to the prediction. So which is like two positive. Okay, so it's having a little bit inaccuracy. And now let's check the other accuracy metric. So sensitivity, specificity, precision. Okay, so we can see that it's actually they are close to 1, or which is considered quite good. And let's say we want to do any optimization, we can try to play around with the parameter here and to make it more perfect. Okay, so we can now take a look at the genes coefficient that the elastic net select to build its model. So this is GOEF is the coefficient. Okay, and 24,944. Some of them show that it's actually just zero coefficient value. So we just want to get to those that showing some coefficient value. So we repeat the command to get rid of all those zero value, and we see again. Okay, so now we can see that this is the six of them that contain, but actually the first one is the intercept. So we also want to remove the intercept, and now we can see they are actually having 143 that posting all this coefficient value, and this is going to be used by our next analysis. Okay, and then now we want to get to know the genes. Now we try to get the common gene name from this lemma dash this particular column to get the external gene name. We can check the common gene between the lemma and elastic net result by using intercept function like this. Okay, it's only 2, which means that lemma and the elastic net, they try to get the differentiate express gene in kind of different approach, and they actually some having their own concern, own consideration, and that's why it's having 2 that in overlap. Okay, we can now take a look at how our sample cluster together by running a hierarchical clustering script. Okay, so we get the things all like this. And okay, we look at the gene elastic net found and use it to cluster the sample. Okay, and we highlight that green is, let's say, they are actually overlap with the lemma result, or white that show that not really the overlap genes. So as we can see here, you can see that they're actually having 2 groups. The sample highlighted in red are solid sample, solid tissue normal, and the highlighted in black are primary solid tumor. And here the green color means that this gene is particularly overlapped in the lemma, and we can see that they having kind of like some kind of difference in terms of the gene expression level. And interestingly, the two overlap genes that are also detected by the lemma analysis are only associated with high expression in the control group. Okay, we can now proceed to the gene expression and survival analysis. We have to get the differentiated genes between the two groups in the tumor tissue and normal tissue. So from the lemma RAS data frame, we already have the top differentially expressed gene ordered by significance. As an example, here we just take the first one to analyze. Okay, so this is the first one. The gene name C2ORF88. And we try to get the assemble gene ID, external gene ID. Okay, and now we can just see, and let's visualize how much differentially expressed it is. Okay, quite a big difference, right? So this is the sample with the disease that show the gene always in the lower expression, and the sample that with healthy normal people, they always have a higher gene expression level. Okay, now we want to see if the difference in the expression value do affect the prognosis or not. We have to divide the patient into two groups. One is up and the other one in the down regulation. Okay, and we use the median as threshold to divide the two groups. Okay, so now we have another extra column that is, let's say the patient is if the median value is more than the median value, that's called up, or else it call it down. We also create a new column as shown as the last time, the previous part. We also create the disease column to show true for the dead patient and false for the alive patient, the patient with alive, and also the overall survival to know the days of number of days that the survivor or to the death. Okay, and now we can fit a survival model. As we mentioned earlier, this is a linear model that show the dependent-independent regression model. So we have the dependent variable here, overall survival, disease versus the independent that is about the genes. The gene that we select here is called C2ORF88. Okay, let's have a look. Okay, and we can also get the p-value to see, oh, this is not really a good, not really anything affecting the prognosis, because the p-value is not significant. And finally, we can see it in the plot. Okay, so we can have all the things that we want. We want the p-value, we want the risk table. Also, we put the title here with the gene's name, so we know what is it. We can see that the genes does not appear to make any difference for the prognosis of prostate cancer. So even though we can see such a big difference at the end, at the later day, it is actually represented by very small sample number, which is 3 + 1, 4. And we can see most of the sample, they actually look very similar in gene expression level throughout the time of survival. So we couldn't really diagnose the patient. We couldn't really predict the patient's survival rate based on this gene. So that's all for the analysis. Thanks for listening until now. I know this is such a long video, but this is the best way I could find to explain it to make myself understand, and hopefully this is as detailed to make you to understand the steps and concept. I hope to make more videos like this. If you find this video is informative, please give me a like and subscribe. Thanks and bye. See you next time.